This is Sushma Nethani from Oregon State University. Greetings to you all from Plant Reactome team. Today I'm going to talk to you about Plant Pathway Portal of Grameen. You can access Plant Reactome from Grameen homepage. You can also search for genes and pathways right here, or you can go to plantreactome.grameen.org and access Plant Reactome homepage. In Plant Reactome, we use rice as a reference species for pathway curation and use gene ontology-based methods to project pathways in other 74 species. Here's a quick tour to pathways. You can browse pathways, and here is a pathway browser view. By default, we have reference species Rhiza sativa here. Nonetheless, you can use this pull down button here and select your favorite species including maize. On this left side panel, we have listing of pathways. Using this plus and minus sign, you can navigate from any reaction level up to the super pathway level. Depending on the selection that you have here on the left panel, you can have a diagram in this particular window. So this is a top level fireworks hierarchy view of various pathways. Another example is a detailed pathway view of a genetic regulatory pathway that is involved in inflorescence development. You can see we also have metabolic pathways, hormone signaling pathways, transport pathways as well. On this bottom panel, we see expression of the genes that are associated with this pathway, and this data is being projected from EBI Atlas project. Recently, we are able to also project gene-gene interaction data from BAR and from a couple other sources. A quick view of maize pathways. If you select maize here, then accordingly you can see the projected pathways for maize. We provide expression data also for maize. We also provide links to the gene pages, etc. However, we don't have any gene-gene interaction data right now for maize. So if you'd like to share any good data with us or direct us to a good publicly available gene-gene interaction data, we will be happy to host it for maize community. This is really a cool example of gene-gene interaction data that is coming from ARANET and being projected on plant reactome pathways. This tool and this utility really complements our curation efforts, which are relatively slow, and by this utility we can provide more nodes and more genes that are providing context of plant pathways to our users, and our users have more power to form a data-driven hypothesis for their experimental research. What else you can do? You can bring your proteome, metabolome, or gene expression data set. This is an example of gene expression analysis from RNA-seq data. As you can see that expressed genes are mapped to the pathways, and you can see boxes green to yellow to red depending on expression. And if you click on these enzymes that are depicted inside these boxes, you can see mapping of the homologous genes that are mapped here, and you can see their differential gene expression. This is really powerful to identify candidate genes. You can query our data using API. We have really a cool widget that is available on Grameen homepage. You can also embed it in your your web page if you like to. I like to bring your focus on Pathway Jamboree that we are hosting alongside the ICPO conference from August 7 to 10. We have travel fellowship available for Jamboree participants. So if you are into pathway curation or are helping us to identify good data for interactome overlays, please apply. Deadline for application is 30th April 2018.